The Sanger sequencing method is one of the most widely used methods for sequencing DNA. It was developed by Frederick Sanger in 1977 at the University of Cambridge. This process largely relies on the principles of DNA replication. The first step is denaturation and annealing. A double-stranded DNA molecule is separated by heat. And an RNA primer, indicated by the yellow box, is annealed to one of the strands, which acts as the template strand for the process. Here, it is indicated by the blue strand. Next, 4 test tubes are assembled, and into each of them, one of the 4 types of dideoxynucleotides, also known as ddNTPs, is added. Then, into each of the 4 test tubes, DNA polymerase is added. A mixture of radiolabeled deoxynucleotides or DNTPs is added. A single-stranded DNA with the RNA primer annealed to it is added as well. The radiolabeled DNTPs will lead to the formation of radiolabeled oligonucleotides at a later stage, facilitating autoradiography. In each test tube, DNA chain synthesis happens complementary to the single-stranded DNA template. However, chain synthesis is terminated as soon as a ddNTP is added to the growing chain. This is because it lacks a 3' hydroxyl group, which means the 5' end of the incoming nucleotide cannot attach to it. The attack of ddNTP to the DNA chain is completely random. As such, depending on when a ddNTP adds onto the growing chain, oligonucleotides of different lengths are produced. Now, we want to separate the DNA fragments of different lengths of each tube, and to do so, we use a procedure called gel electrophoresis that uses a polyacrylamide gel specifically, because it has smaller pore sizes than agarose gel. Thus, it has a very high resolving power and can separate polynucleotide chains that differ in length by only 1 nucleotide. The gel is placed in a buffer that has ions that can carry current. The gel contains indentations that are called wells, where we inject the DNA fragments of different lengths that we created in each tube. Then, we apply an electrical field that has a negative pole on the side of the gel with the wells and a positive pole on the other side of the gel. Note that the DNA is negatively charged due to the phosphate groups in its backbone. So when the electric field is applied and the current is running through the buffer, the negatively charged DNA fragments move away from the negative pole to the positive pole through the polyacrylamide gel. And the larger the DNA fragment is, the slower it moves. Thus, the fragments get separated based on their size as they travel. The contents of each test tube are gel electrophoresed. The oligonucleotides, which would be radiolabeled, separate depending on their molecular weight and appear as distinct bands during autoradiography. These bands show the oligonucleotide which differ in length by 1 base pair, allowing them to be compared. We would know the identity of the terminal base of each oligonucleotide as we had added a different type of ddNTP into the 4 different test tubes. In this manner, By comparing the sizes of the oligonucleotides, we can determine exactly which nucleotide follows the one before it. Hence, we have obtained the sequence of the DNA chain complementary to the single-stranded template utilized in this process.